next speaker is Linda Richards. Linda Richards is a leading developmental neurobiologist whose discoveries have defined the fundamental mechanisms regulating the wiring of neuronal connections between the two hemispheres of the brain.
Thank you very much, and I'd like to begin by thanking the members of the Academy for election as a Fellow. And on this occasion, I'd also like to thank my mentor, Professor Perry Bartlett, and my family for their support of my career. So I'm interested in how the brain becomes wired up during development, and I hope to convince you today that the brain actually does want to wire in order to function in the adult. And I became interested in this topic in my second year of my undergraduate degree when one of my lecturers, John Nelson, actually introduced this topic of brain wiring to me and then encouraged me to go on in this field. And later in my career, I felt that second year of university was really quite late to have first been introduced to the field of neuroscience, which is one of the most exciting fields of science, I think, that's possible for people to work in now. And so I started a competition for high school students to learn about the brain and to give them some inspiration to perhaps pursue a career in science and to understand why scientific research is so important for the Australian community. So I find wiring of the brain a really fascinating topic because it underpins all of the functions that the brain is capable of. And we can see brain wiring at a very gross level by magnetic resonance imaging. And a little bit later, I'll be showing you some images of some quite sophisticated brain imaging that we can now do. Another reason that I'm very interested in how the brain is wired is that we think that deficits in brain wiring may be associated with human conditions such as autism, schizophrenia, intellectual and motor and sensory disabilities in children and in adults. So in order to give you a little bit of background on what's going on during brain wiring, there are two main types of cells in the brain, both neurons and glia play a very important role in the formation of brain wiring. So when we think about brain wiring, sometimes we might think that it's hardwired and predetermined. And I want to show you today that, in fact, during development, it's a very plastic event. In the adult, there's, of course, synaptic plasticity going on. But what I'd like to talk about today is long-range axonal plasticity, which is very much dependent on the role of glia within the environment and the molecules that they express, and other events that occur which enable neurons to find their target, their correct target, out of the 85 to 100 billion neurons that are in the brain. So how is that possible? So in order to study how the brain is wired up, my laboratory focuses on the formation of the largest fibre tract in the brain, which is called the corpus callosum. And you can see it here. It connects the two cerebral hemispheres. And in these images here, this is an adult human brain with the dorsal part removed. So you can see this massive fibre tract connecting the two hemispheres. Now, we know a bit about what the function of the corpus callosum is from work done by Roger Sperry, who won half a Nobel Prize in 1981 for his work on split brain patients who'd had their corpus callosum surgically cut to control epileptic seizures from moving from one hemisphere to the other. And what he found was that people who had had this surgery were unable to properly integrate the information coming into the two hemispheres of the brain. But there's another group of individuals that have congenital malformation of the corpus callosum, and I would suggest that these people are actually even more interesting to study. So corpus callosum malformations occur in about 1 in 3,000 live births, and it can be found in isolation, that is, only the corpus callosum is malformed, or it can be associated with over 300 human congenital syndromes. And so here is an individual with complete absence of the corpus callosum where the fibres have not crossed the midline here. So in amongst this whole group of individuals, the phenotype falls into three categories: very high functioning people who may have deficits in complex social interaction, a more moderately affected group that may have language developmental delay, sensory or motor deficits, and then a very severely affected group of children that may never learn to walk or talk. So in my lab, we use a number of different species to try and understand the mechanisms involved in brain wiring. And this is a coronal section through a mouse brain with the corpus callosum axons labelled in green. The cell bodies are sitting over here. They send their axons across the midline into the contralateral hemisphere where they find their targets. So today I'd like to talk about two aspects of our work. Firstly, on cues that are found at the midline that are involved in guidance of the corpus callosum, and then some of our newer work looking for cues that are involved in contralaterally projecting axons finding their correct place in the other hemisphere. So to begin with the midline guidance cues, I mentioned that glia were very important for wiring the brain. And what you can see here, so this is a high power view of this boxed region here, and the red axons are the corpus callosum, mouse corpus callosum axons, and the glia are labelled in green. And you can see the glia completely surround this tract. And this association between the glia and the axons is highly evolutionarily conserved. All animals with a nervous system have glia at the midline that are involved in formation of commissural projections, so axons that are crossing the midline. And so, and also throughout the whole neuroaxis, both the brain and the spinal cord. So we've been studying a group of transcription factors with our collaborator Rich Granasowski called the nuclear factor 1 genes, and these are absolutely essential for the formation of midline glia in both humans and in mice. We've also described many of the mechanisms that are involved in actually guiding these axons during development, and the role of these ligands that actually are secreted proteins that attract the axons towards the midline and repel them up and away. They're able to sense these through the expression of these receptors. And again, these ligands and receptors are highly evolutionarily conserved. Now, moving on to the second aspect of the work I was going to tell you about today, how neurons find their targets in the contralateral hemisphere. So by now, the callosal axons have already crossed the midline, but they need to find their correct target in the contralateral hemisphere. And for this, our model system is the mouse somatosensory or touch system, where the somatosensory information in the rodent is encoded in a topographic manner. The whiskers on the face are perceiving the environment, and that information is faithfully transported up to the contralateral cortex. And you'll notice that this is a fully cross projection in rodents. So the first time the two sets, I'm only showing one side of the innovation here, but the first time the two sets of information are brought together is at the level of the corpus callosum. So in order to try to understand what's going on here, I should say that the callosal axons are crossing the midline around just before birth and just after birth in the rodent. So they're actually targeting this region around the first postnatal week. So once the animal has started interacting with its environment. And we already previously knew that activity was important for the wiring of synapses, but more recent work had shown that if you remove the whiskers on one side of the face, then this targeting event doesn't occur, and this is quantified here. What we found is that if we remove the whiskers from both sides of the face, that in fact we got a partial rescue, shown here in the blue line, of this event. So this is the unilateral removal, the bilateral removal, and the control. So it's not a full rescue, but it is a partial rescue. Now, this was puzzling to us because we knew that neural activity was important for the formation of this projection. And so we hypothesised that maybe it wasn't the level of activity that was important, but in fact whether the inputs were balanced in a way that the callosal axons were looking for similar information in both hemispheres. And so to address this, we removed the whiskers in different patterns on the face of the animal. So the same number of whiskers, but in either a symmetric or asymmetric pattern. And we found that we only got the rescue if the whiskers were removed in a symmetric manner. So I'd like to just talk about where this work is going and how does this inform what's happening in the developing human brain. So we now know that all brains are not wired the same, and we've known for a long time that people with a genesis of the corpus callosum have ectopic tracts. This one in particular is called the Probst bundle. It was identified by Probst in 1901, and it's shown here with a newer form of magnetic resonance imaging, diffusion imaging, with tractography performed on that. So just recently, a very exciting development has been the formation of an international consortium for the corpus callosum and cerebral connectivity. We're calling ourselves IRC5, and these are some of the members here that were at a meeting just in March at the Queensland Brain Institute. I've also been privileged to work with AusDoc, the Australian Disorders of the Corpus Callosum Patient Support Group, and I'll leave you with a very interesting subject that's recently been working with us. So this subject has the middle part of their corpus callosum missing here, and we don't actually understand why that region is missing, but something that we're working on. And you're looking at a T1 or anatomical image here tilted to the side so that you'll be able to see the fibre tracks in the other hemisphere. So if we perform tractography here, what we would have previously predicted was that those parietal axons would not be connecting. So the parietal cortex would not be connected, and here these would be the yellow axons. And what you can see here in this subject is that, in fact, they're crossing via the anterior commissure, and the parietal cortex is actually still connected up. So the brain really does undergo long-range axonal plasticity, and I'll close there by acknowledging my current lab members, my former students, my collaborators, and I'd also like to acknowledge the fantastic imaging facilities at the University of Queensland that are NCRIS funded, and also our funding. Thank you very much.
Thank you very much, Linda. Questions? Eugenie.
I might be wrong, but isn't it the monotremes don't have a corpus callosum? How are they wired up?
Right, so in fact, the corpus callosum is an evolutionarily newer structure, and only placental mammals have a corpus callosum. And so both monotremes and marsupials don't have a corpus callosum. They do have a 6-layered cortex that is connected via that anterior commissure. So the commissure that was utilised in the last subject that I showed you, that is the commissure that marsupials use, and monotremes.
Roger.
Not a question, but it's a comment. A recent paper 2 years ago from the University of Perth, West Australia, by Foteini Hasiotu, published in the Journal of Human Lactation, showed that human breast milk contains embryonic neural stem cells. And a paper just published in The Lancet last month from Brazil, a clinical trial of 3,000 women, about half of whom were breastfed and about half of whom were bottle-fed, showed a highly significant increase in income and intelligence of human babies who had been reared on breast milk. Do you think that this could be a major route of transmitting maternal neural tissue into the newborn?
Well, that's a very interesting question. I find it difficult to believe that they would pass out of the nipple and into the circulation, and then via the blood-brain barrier into the brain. But so I think that the second study that you talked about in terms of nutrition, it's very evident that sensory stimulation, intellectual input into the developing baby is incredibly important for the final wiring of the brain into a functioning. I mean, the baby has to interact. It has to function in order to allow the brain to wire up correctly. And so better nutrition is also going to play a role in that.
That food for thought. Thank you very much.